All right, so thanks, Royal. So in this talk, I'll be giving a very selective tour of the human nervous system. Now, I say very selective because the topic is so large that only a tiny fraction of the nervous system can be covered in the short time available. And I'll be dealing with the human nervous system as I know it best. And in terms of what it can do, it also appears to be the most functionally complex. However, a lot of the arguments for the design of the human nervous system apply also to the nervous system of animals. Regarding the mind, I regard it as distinct from the physical brain. Obviously, though, the two go together. And so when talking about the brain, it may also include the mind, depending on the context. Having already been given an intro by Royal, I'm just going to quickly skip through a bit about my background. And as Royal mentioned, I've got a background in recording brain electrical activity, and my doctoral project looked at was looking at brain activity associated with hearing, auditory hallucination, and schizophrenia. And the above shows a sort of animation of the EEG, the electroencephalogram, and also an electrocap used that you put on a subject's head. And so I looked at a rather specialized form of EEG recording called a steady state visually evoked potential, but I'm not going to go into any more details about the technique for time reasons, as my talk is not really about that. However, the end product was nice colored maps, or they were at the time, and the editors of a paper from my doctoral study liked the maps, and they put them on the cover there, although the brain doesn't actually look like that, but just joking there. So yeah, I thought it was rather nice at the time. And yeah, as Royal mentioned, I've been doing over a decade in EEG related neuroscience research. Before I started teaching at the university I was working at, and for a decade or so, I taught in the biomedical sciences, as mentioned, focusing on neuroanatomy and skeletal muscle physiology, among other subjects. Yeah. And also in the last 15 years, as Royal mentioned, I've had an interest in paleoanthropology from a creationist viewpoint. Now, one reason for that is that it seemed to be an area where evolutionists could more easily intimidate people into believing their story by, for example, just showing them fossils alleged to be of ape men, and most people may not be in a position to kind of judge whether that really is that or not. So that kind of got me interested in that area. So I've written a few articles in the Creation Ministry International's Journal of Creation, and also on a web for them, and also in their magazine. And I still write for them and associated with them. Most recently, I was part of a book project with Jerry Bergman, where in my 10 chapters, I analysed many of the alleged ape-man fossils like the Australopithecines, Homo habilis, Homo erectus, Homo floresiensis, and more. However, that's not the topic of today. Now, if you want powerful evidence for a designer's existence, then look no further than inside your head. Now, whilst it may not be that impressive to look at, the human brain, weighing only about 1.4 kilos or 3 pounds on average, is often described as the most complex arrangement of matter in the known universe, and with good reason. It makes machines created by man, as great as some of those accomplishments are, seem trivial in comparison. As an example, consider that the 3D world we experience is essentially a construct by our brains. So every awake second, environmental signals of one kind or another, such as light, sound, touch, temperature, and airborne molecules conveying odors, are converted into electrical currents of iron called action potentials by sensory receptors in the skin, or special senses like olfaction or vision and hearing. So in the above diagram here, you can see this is a compound action potential because it's the combined electrical potential or voltage of a group of nerve fibers in a single nerve. So this is a sciatic nerve of a cane toad, and basically it contains hundreds of nerve fibers, so it's not just from a single nerve fiber. And so and this apparatus here looks like one of my setups. And so from memory, you apply the voltage here to the nerve across. You can just not barely see there, and you record from here. Anyhow, moving on. So these are ionic currents which bear no resemblance to the information they carry, travel into the brain via nerve fibers, cables, if you like, illustrated in the diagram above. Now, the two optic nerves, these two optic nerves there, each contain roughly 1 million or so nerve fibers, and they become optic tracts once they sort of pass through the optic chiasm, which is where about roughly half of the nerve fibers cross over from each eye, and the optic tracts then travel to the lateral geniculate nucleus of the thalamus, where they synapse on neurons there, and then these neurons then go to the occipital cortex, the primal visual cortex there, and basically for further processing. Now, the above illustrates how intricate, yet also how orderly the brain's hardware is wired with nerve fibers, like an integrated circuit board, for want of a better analogy. Now, once received, the brain somehow decodes the information and processes it so that the original signal can be presented to you, the ghost in the machine, or mind or soul. I don't want to get into splitting those things up. I just usually just refer to as the mind. And all this brain processing occurs automatically, without you having to think about it. Rather, you can think about what you see, or what you hear, smell, taste, or feel in the case of input to the brain from other sensory nerves. And this allows you to see the world in vivid colour, hear the sound of a waterfall, experience the heat of a hot summer's day, feel the breeze impacting on your skin, smell and taste food, etc. And you experience all these sensory modalities simultaneously in an integrated way in essentially real time. And I say essentially real time because action potentials travel at a finite velocity up to about max of about 120 meters per second, which is in large diameter axons. Now, no one knows how the brain does this, but it surely involves signal processing orders of magnitude beyond anything we got. Is it really feasible for an unintelligent process to have done this without plan or purpose? Now, although essentially a continuum, for convenience, the nervous system is divided into parts, regions, and systems. So the brain and the brain and spinal cord form the central nervous system. So that's kind of, obviously most people know that. And then extending from the from the central nervous system is the peripheral nervous system, abbreviated PNS here. So you can see all the nerves extending from the spinal cord here. Now you can divide into systems, some sort of kind of more arbitrary than others, but essentially, so you have a peripheral and central nervous system. You have in the central nervous system a brain and spinal cord. The brain then can divide it into the cerebrum, cerebellum, and brainstem. The brainstem divided into midbrain, medulla, and pons. And basically, the cerebrum consists of a telencephalon, diencephalon. And then you have the peripheral nervous system can be divided into an efferent, which is an input division, and an efferent output division, or a motor division, as it innervates muscles, whether it's skeletal muscles, whether it's somatic motor division, or whether it's the muscles in smooth muscles in blood vessels or in other organs of the body, or in the heart, it may innervate muscles there, or in glands, or even adipose sort of fatty tissue cells. And this is known as the autonomic nervous system here. And then you have the afferent infant division, which is the somatic sensory. So that's all the senses on your skin, whether it's for hot, cold receptors, or pain, or touch, things like that. And also there's receptors in your muscles, which basically you got your muscle spindles, things like that, and it's in your joints as well. And then you have the visceral central division, which is there's nerve fibers inside your organs, which monitor your organs, or inside blood vessels and things like that. And then you have the special sensory receptors, which is your eyes, your ears, and things like that, which gives that sort of information. All right. Now, so concerning the autonomic nervous system, which is part of you have basically it consists of, as mentioned, efferent divisions that carry out motor commands, and many of the functions under its control are self-regulating or autonomous. It contains neurons that innervate the heart, glands, and the smooth muscle internal organs and blood vessels. Now it's divided into 3 divisions. 2 of them are shown on the screen there: the parasympathetic and sympathetic. I'll just briefly discuss them, and then just mention the third one. Now, so you have the sympathetic division. It inhibits or opposes, in generally, the physiological effects of the parasympathetic division. So basically tending to reduce digestive secretions, speed up the heart rate, dilate the pupils, and cause contraction of blood vessels. And the parasympathetic, in turn, tends to inhibit or oppose the physiological effects of the sympathetic nervous system. So basically tending to stimulate digestive secretion, slow the heart rate, and constrict the pupils. But it has no apparent effect on blood vessels. Now there is a third division, so sort of lesser well known, and called the Interactive Subdivision, and that is a collection of neurons in the gastrointestinal tract that constitutes the brain of the gut, as it's sort of called, and can function independently of the central nervous system. It is influenced by the sympathetic and parasympathetic systems, though, but it also has independent control, that is local reflexes. And in the diagram here, you can see the parasympathetic and sympathetic innervation. So the sympathetic innervation from the thoracic lumbar region, you have the cranial nerves emerging here. And basically you can see all the, not the cranial, the spinal nerves emerging here, and innervating all the organs. And you can see the parasympathetic division from the sacral region, the spinal nerves innervating organs, and then they have the cranial nerves up here innervating certain organs. Okay. And so many of the self-regulating or automatic functions of the autonomic nervous system are controlled by nuclei in the brainstem. So basically, in this kind of region here, so this is the pons here, medulla here, the midbrain here. And so they're controlled by nuclei in the brainstem, as well as nuclei in the hypothalamus of the central nervous system. And now nuclei are a cluster of neurons in the central nervous system outside the cerebral cortex of the cerebrum and the cerebellar cortex of the cerebellum. In the peripheral nervous system, nuclei are called ganglia. So basically, if you look, these are ganglia here. So in the parasympathetic innervation here, the ganglia, part of the peripheral nervous system, are located closer to the organs, so they're clusters of neurons here. Whereas in the sympathetic division, the ganglia are kind of more closer to the spinal cord, further away from the organ. So that's kind of a different thing there. Now, so for example, within the nucleus of the medulla, you have cardiac and respiratory reflex centers involved in controlling things such as heart rate and blood pressure and basic pace of respiratory movements, that is inspiration and expiration reflexes. Further up the brainstem in the pons, there are respiratory centers which modify the respiratory centers in the medulla. So this is quite complex, and some of these functions are very vital to life. Now, these are all part of the reticular formation, so that a group of more than 100 small, loosely associated nuclei spanning the central core of the brainstem from medulla to midbrain, that although poorly understood are involved in all sorts of functions, including motor control, sleep and alertness, pain modulation, etc. In addition to the things I just pointed out, the blood pressure, heart rate, respiratory, expiration, inspiration. So this represents here the reticular formation here. Now, I said in the first talk, I didn't know what the language was that, and Boris told me, but I couldn't work out what he's saying, so he may be able to tell you what language that this is written in. But it was kind of the best diagram I could find in Wikimedia Commons. So anyhow, thing is, how did all these nuclear centers or groups, some wider to life, get organized without planning or design? By the supposed evolution mechanism of natural selection sorting random mutations, what would be the waiting time to create all these nuclei and the target cells and organs, as well as their integration? I suggest forever. Now, one can ask similar questions with about the organization of the hypothalamus and its many nuclei, which is shown in a diagram here. So all the nuclei here, some are listed here. Put the names. I'm not going to go through and name them. But the hypothalamus is the main control center for both the autonomic nervous system and hormonal, that is the endocrine system, as well as a center of integration between the two. So it's a very, very busy sort of center or structure. Now, the hypothalamus controls hormone production in the body through its control of pituitary secretions, the pituitary gland. It helps to regulate temperature, metabolic rate, water balance, appetite, and influences emotional behavior. It regulates reproductive development and function. Now, the above shows the major hormones or factors in the hypothalamus. Now, this OT should be oxytocin. It should be here. It's produced in the hypothalamus. I don't know what. Anyhow, that's all right. Now, so basically, and it also shows the hormones and stuff that are manufactured in the pituitary gland here. These are called the releasing hormones produced in the hypothalamus, or releasing factors. So whether you consider them real hormones or not, but basically they're released into the anterior pituitary gland, the adenohypophysis here, and basically they trigger the manufacture and or release of the pituitary hormones here in the anterior pituitary gland, and then these hormones then may target cells, or they may target another gland, which stimulates another hormones to be produced. Okay, in the other setup here, you have these hormones, oxytocin and antidiuretic hormone, basically also known as vasopressin. But these are produced in the hypothalamus, and they travel down the axons into the posterior pituitary gland, the neurohypophysis, and they're released into here in the capillaries here and go into the systemic circulation here. So that's kind of the sort of release mechanism. I don't know what happened here. I think I moved something. Okay, we've got the right diagram now. Okay, so this here is an example of the kind of feedback control of endocrine secretion by the hypothalamus. And sort of this example is by negative kind of feedback. So for example, in response to stress, the hypothalamus releases the corticotropin releasing hormone CRH here. So basically, in this area here, and that so sort of is released into the anterior pituitary gland, and then basically that stimulates the production and a release in the anterior pituitary of the adrenocorticotropic hormone ACTH. And that step is actually emitted, so it's another step. So you can put it in here. Okay. Now the ACTH then stimulates the generation of glucocorticoids, which is known as cortisol in humans. So basically that's sort of produced in here, and that's produced, the cortisol is produced in the cortex of the adrenal gland, which sits on top of the kidneys. And then this is kind of released into the blood. From there, by the way, the glucocorticoid hormone, the cortisol, is known particularly for its anti-inflammatory effects and in immunosuppressive actions. Anyhow, the increased level of the cortisol hormone is sensed by the hypothalamus or perceives as a normal concentration, and then basically it stops the release of the CRH hormone, the corticotropin releasing hormone, which kind of stops the cortisol from being released into the bloodstream, but once it gets a low level again, it has sensors, nerve receptors in the blood vessel. It detects there's a low concentration, and then it ups the production again. So you have this negative feedback loop here of regulating hormone production, which is sort of great engineering, particularly as it must control multiple hormones simultaneously. All right, so now we have basically a bit about the nervous system, the central nervous system. I mean, so there's close to 100 billion neurons in a central nerve, in the human brain. I think the current estimate is about 86 billion by some. Whether that's, I don't think that's the end of the story. Probably hear another estimate in a few years. That's compared to about 30 trillion cells in the body. And there is likely as many neuroglia, that is, supporting cells in the human brain, as nerve cells. Up until recently, it used to be said that there are about an order of magnitude more neuroglial cells than nerve cells in the brain. But this figure is apparently wrong. I only discovered this while doing some checking whilst preparing for this talk. Not sure that anyone knows what the correct number is, though. There's also more than 50 types of nerve cells, over 300,000 motor cells innervating skeletal muscle. There's several million sensory cells supplying information to the central nervous system. An average nerve cell has several thousand synapses with other nerve cells. So that means there's trillions and trillions of connections in the brain. So it gets very complicated network. Now the brain is it's suspended inside the cranium and floats in cerebrospinal fluid. So that, as a protective mechanism, helps cushion it against shock and things like that. It's also heavily fortified, being encased in bone, that is the skull and spinal column. It's also protected by the blood-brain barrier, which stops certain substances passing into the central nervous system from the rest of the body. So the blood-brain barrier consists in the ventricles. You have the choroid plexuses, which is one part, but also the capillary networks also act as a blood-brain barrier. Now, so the central nervous system, as mentioned earlier, it consists of the cerebrum, which consists of the telencephalons, the two hemispheres, and the diencephalon, and also the cerebellum and the brainstem here. You can't see the midbrain. There's a pons medulla and a spinal cord, which kind of is down here somewhere, sort of arbitrary where the medulla begins and the spinal cord ends. It's sort of at the opening of the skull at the forearm and magnum position, sort of where a kind of spinal cord starts. And you also have a fifth lobe of the brain, but you can't see it from here because it's sort of in this, you need a sagittal section to see it. It's called a limbic lobe. You have the central sulcus, which separates the frontal lobe from the parietal lobe. It also separates the precentral gyrus from the postcentral gyrus. So this is where the sensory information comes from, the somatosensory from the skin and things like that. And this is a primary motor cortex, which when motor movements are planned, whether it's in a premotor cortex or supplementary motor area, whether it's based on visual cue or memory, the information is then sent to the primary motor cortex to carry out the commands or actions. So that's it here. Now, so the cerebrum, as you can see, is the largest and most prominent structure of the brain, and it's divided into paired cerebral hemispheres by the longitudinal fissure, as you can see there in the red in the animation on the screen. And together, as already mentioned, the two cerebral hemispheres are known as the telencephalon, which is the cerebrum minus the diencephalon. So the diencephalon is situated just above the brainstem, and the two of its main structures are the hypothalamus and the thalamus. Now, the hippocampus is something that the telencephalon differentiates into the hippocampus. You can see these yellow structures here. So they're involved in memory. Now, the hippocampus is an extension of the cerebral cortex, which has curled up in the inner medial region of the temporal lobe. And it consists of three distinct layers of cortex. The hippocampus appears critical for the consolidation of long-term memories, with the strengthening of synaptic connections between nerve cells thought to be how long-term memory is stored. And long-term memory does not appear to be stored in any specific region, but it is spread across the brain. How exactly the strengthening synaptic connections allow information to be stored or encoded and later retrieved is a mystery. Also, hippocampal neurons possess a very low absolute threshold for convulsive discharges. So the hippocampus is of special importance for the origin of epileptic seizures and memory deficits. The telencephalon also includes the basal ganglia, so these structures shown in red here, which consists of several large nuclei embedded deeply in the white matter of the cerebrum, as well as extending to the or in the cerebral hemispheres, I should say, as well as extending to the diencephalon, and also the midbrain, so basically the midbrain is here, and the basal ganglia are involved in regulation of stereotyped automatic muscle movements and muscle tone. Now you have the cerebellum is positioned behind the brainstem and is involved in in motor coordination. It is often called the mini brain, and that's the reason for that is it has its own, as alluded to earlier, as like the cerebrum has its cerebral cortex. The cerebellum has its cerebellar cortex. It's sort of grey matter on the outside. It's three-layered. Whereas the cerebral cortex, most of it, not all, but most of it is six-layered. But it also has a white matter embedded in the center, but in the center it has these nuclei as well. And basically all outgoing information from the cerebellar cortex will first seen at these nuclei, except for some from that comes from a part called the vestibular cerebellum or the floccular nodular lobe, which has a lot to do with balance, which send information, which kind of the Purkinje fibers can project directly out to the vestibular nuclei in that case. And the Purkinje fibers, they have dendritic trees, which is where the input comes from motor neurons. They can have up to about 200,000 synapses on it. So these are the largest sort of dendritic trees of any nerve cells in the nervous system. Okay, and one of its main the cerebellum is an automatic processing center. One of its main functions is the regulation of movements, underlying posture and equilibrium. It maintains equilibrium, that is balance using information from the vestibular apparatus of the inner ear. Another main function is the programming and fine-tuning movements control at the conscious and subconscious levels, such as the learned movement patterns. Think of a gymnastic routine, you know. And if the gymnast has to think too much about it when they're doing it, they're probably going to fall on their face. So this is where the cerebellum comes in. You know, the memory is stored there. Okay. Now, the corpus callosum is the white matter here. Okay, so that's known as the great commissure of the brain, and the left and right hemispheres of the cerebrum are connected by the corpus callosum, which is a massive bundle of commissural fibers. These are axons that connected to hemispheres of the brain. These are not the only commissural fibers. There are others as well, but they're nothing of this size. So this is the major ones that interconnected to hemispheres. Okay. So I already mentioned the brainstem here. You have the midbrain here. You have also here it's known as mesencephalon. The pons and myelencephalon is the medulla. The medulla spinalis is the spinal cord. You have the superior and inferior colliculi, and they're involved and play an important role in controlling saccadic eye movements and hearing, respectively. And the hypophysis is the pituitary gland. The epiphysis is the pineal gland, and also the thalamus that is one of the major parts of the diencephalon. So we'll look at that next. Okay, so you have the thalamus is a paired structure. You can see here. So a lot of the structures in the brain or the central nerve system are paired structures. Now, the thalamus is as a main relay station of information, as a main relay station of information to the brain. All sensory pathways except for olfaction travel to the cerebral cortex via the thalamus. Now, olfactory nerves travel directly into the telencephalon. So basically, all other sensory pathways go directly to the cerebral cortex via the thalamus. Also, motor pathways, for example, from the cerebellum and basal ganglia, also travel to the premotor and primary motor cortex areas of the cerebral cortex via the thalamus. So it's a very busy relay center. So when you're awake, it literally handle millions of signals per second or whatever. Or I don't know how, probably a lot of information, and it's got to send it to specific regions and things like that. Now, based on their locality, there are 6 groups of nuclei in the thalamus, and some of these are further subdivided. So I'm not going to go into and name all the nuclei and things like that. But examples of nuclei from the lateral loop are illustrated in the slide. So these are these orange-yellow things here, as well as these red things here, called the medial geniculate body and lateral geniculate body. They're nuclei. Sometimes they call them bodies for some reason not sure about. Okay. Now, examples of nuclei from the lateral group, which are illustrated on the side, includes the medial geniculate nucleus or body, as mentioned, and this relays auditory information from the inferior colliculus to the primary auditory cortex. Now you have the lateral geniculate nucleus, so the other red blip there, here. That gets input from the retina and relays the visual information from the primary visual cortex, to the primary visual cortex. You have the pulvina here. So that basically projects visual information about eye movements from the superior colliculus to higher order visual areas. So the higher order areas means they're not the primary visual cortex. The higher order visual areas lie outside, close by to the primary area, but they do process-specific aspects of the visual information. Now, based on the extent and function of their cortical connections, the thalamic nuclei are also divided into two major functional classes. So you have relay nuclei and diffuse projecting nuclei. Now, the relay nuclei project to specific regions of the cortex. For example, visual information from the retina is relayed to the primary visual cortex by the lateral geniculate nucleus, as already mentioned. Now, the diffuse projection nuclei, for example, the interlaminar nuclei, project to wide regions of the cortex believed to be involved in arousal, that is being alert, awake, and attentive. Now, according to Nicholas Sheridan and Prasanna Tati, the thalamus can divide into approximately 60 regions called nuclei. Each nucleus has unique pathways as inputs and various projections as outputs, most of which send information to the cerebral cortex. How could this setup be designed without deliberate intelligence engineering? Would you build a computer hubs or telephone exchanges by potluck? Companies know better. For one thing, millions of wires being wrongly connected would be very bad for business. How could such an immensely functionally complex relay station and brain like the thalamus be thought of as having been built without intelligent design? It is illogical. All right. Now, this diagram shows it shows the sort of position of the grey matter and the white matter. It also shows the sectional planes, which is when you're talking about the anatomy of the brain and you're dividing up in terms of locations. You talk about the frontal plane, also known as the coronal plane, sagittal plane, or transverse plane. I prefer it to call it the horizontal plane, but that doesn't really matter much what you call it. Now, the outer quarter of the cerebrum is the grey matter, and it's also known as the cerebral cortex, which is the surface covering of the cerebrum. So the grey matter is the darker colour, although when you're in staining diagrams, the grey matter stains light and the white matter stains dark. So sometimes that can be confusing unless you realise it. Now, the grey matter is a darker coloured tissue and is composed mainly of the cell bodies and neurons, branching dendrites and glial cells. This outer layer, most of which is 6-layered, is highly folded. Now, the white matter is composed mainly of myelinated axon and makes up most of the inner core of the cerebral hemispheres. So grey matter makes up about 40%, whereas 60% is white matter. This shows, again, as a frontal section that is in the frontal plane, okay? Now, gyri are the raised ridges of the folds in the cerebral cortex, and sulci are the kind of the grooves between the gyri. Now, if the grooves are deep, then they may be called fissures, like the longitudinal fissure. Now, only about 1/3 of the cerebral cortex is exposed on the surface. Hence, about 2/3 of cortical surface area faces the sulcal spaces. Now, the buckling and folding of the cerebral cortex, creating gyri and sulci, allows a greater surface area of the cerebral cortex to fit within the confined space of the cranial cavity without having to increase the thickness of the cortex. So you can see particularly the buckling here, this particular area here. Now, 95% of the cerebral cortex contains 6 layers. This cortex, called neocortex, include that which is visible on the external surface of the brain. So you can see here in this diagram here, it illustrates the 6 layers 1, 2, 3, 4, 5, and 6 divided into 6A and 6B. And the other 5% of this, the cerebral cortex is termed the other cortex, the 95% being called the neocortex, which includes that which is visible on the external surface of the brain. The other cortex, the other 5% is buried deep within the temporal lobe, like the hippocampus discussed earlier. Now, the other cortex has very number of layers, but it's always fewer than 6. Now, the cerebral cortex varies in thickness from about 2 to 4 millimeters. Okay, so this 2 to 4 millimeters, and it tends to be thicker in motor and association areas and thinner in the primary sensory areas. The thickness of the 6 layers in the neocortex also varies. So these layers are not all the same thickness. You can see that varies here. So layer 6, but they're kind of different. And also the density, size, and types of neurons within each layer also kind of varies. So you can see here, the layer 5 that has large pyramidal neurons. So these are the only neurons that exit the cerebral cortex. They may project to other cortical areas or to the brainstem and spinal cord and places like that. Now, these ones project to the brainstem and spinal cord, and not so much, not to other cortical areas. Whereas in these layers 2 and 3, the more medium, small-sized pyramidal cells project to other cortical areas. This layer 1 is the outer layer of the cerebral cortex, and that is through nerve cell bodies and nerve fibers. Layer 6 has sort of irregular shaped pyramidal cells which project to the thalamus. Layer 4 is the main input layer of the cerebral cortex. So it tends to have non-pyramidal cells in this region here. Okay. Now, processing within and between the lobes is very complex and involves many different functions. Now, depending on the conventions used, the cortex can be divided into functions that each belong to 50 to 100 separate areas. Now, probably the best known divisions of the cerebral cortex are the Brodmann's areas. About a century or so ago, the German anatomist Corbinian Brodmann identified over 50 divisions of based only on the neuronal architecture of the cortex. Okay, so there's all these regions here. And research has since confirmed or shown that different functional areas of the cortex have a different neuronal architecture. So you can see throughout, whether it's the layers, throughout the cerebral cortex, it's all organized. It's not sort of just a haphazard mixture of things here and there everywhere. It's all specifically organized according to function and purpose, whether you're looking at topographically or within the layers themselves. Now, this diagram here shows how sensory information is relayed to the cerebral cortex via the spinal cord and brainstem. It is all very orderly. So for example, here you have the sensory information is relayed into the spinal cord here. So you have through the dorsal root here, and the dorsal root ganglia consists of the cell bodies of the unipolar neurons with axons project into the spinal cord here. So in this case, you have the axons actually become part of the posterior column here of white matter, which is basically axons, myelinated axons, and form the posterior column, which consists of the fasciculus gracilis and fasciculus cuneatus. So these are basically nerve tracts here, bundles of axons, and they project up to the medulla, where they synapse on neurons here. And when after that, the axons cross the midline and proceed up to the thalamus, where they again synapse at neurons here, a specific area, and then they get relayed to the post-central gyrus to the specific area that they're allocated to here. And this sort of crossing of the midline, both here and in the motor cortex with motor information, explains why the right side of your brain controls the left side of your body and vice versa. And also you can see here some sensory information will synapse on neurons at the level of the spinal cord here. So with sensory information, they usually, it'll be at the sort of the posterior end of the grey horns here, the grey matter of the spinal cord, the nuclei here. So basically you have interneurons here, which is the axons will synapse on, and then these interneurons will then, in this case, it crosses over here, the midline here, and then proceeds up to the thalamus and get relayed to the cerebral cortex. So you have, not only is the white matter is organized, you have the posterior column, lateral column, anterior column here. So you have here is just ascending information. Here, the lateral area of the white matter here is you have both descending and ascending nerve tracts, and anteriorly you mostly have descending motor information, although you have some ascending sensory information as well going upwards. And the grey matter too is organized. So here at the anterior end of the grey matter is basically motor neurons, and posteriorly is more the sensory interneurons that carry sensory information upwards. And these are also organized by functions. So it's all kind of orderly. Okay. Now, this shows how corticospinal pathway. So basically you have the precentral gyrus, the primary motor cortex, sends information here, goes through the cerebral peduncles. These are sort of axons of nerve fibers connecting the cerebrum with the brainstem here. So there's a lot of nerve fibers here. And so basically the information then goes down to the medulla, and then so these are corticospinal axons here. And then in the medulla, these fibers are known as the pyramids because there's bulges on the anterior part of the brainstem or the medulla. And then caudally in the medulla, they cross over and descend into the designated place in the spinal cord here. And when I get to the designated place, then basically they go and synapse. The fibers move anteriorly and synapse on the motor neurons in the grey horn here, and then these project out and innervate the skeletal muscles here. And you also have some 85% may go here, 15% may go descend as the anterior corticospinal tract, in which case they cross the midline at the level of the spinal cord that are designated to, and then synapse on the motor neurons here, and then innervate the skeletal muscles. Now, white matter makes up a considerable portion of the nervous system, connecting numerous different regions in a very orderly way, often involving two, sometimes more synaptic connections en route. How did this order arise without intelligent planning? If it was created by a purposeless process such as evolution, if it were possible, then we would expect more of a spaghetti and meatballs arrangement, not orderly parallel nerve fibers and tracts and things like that, everything being nicely organized, complex but organized, and it is like an integrated circuit board is a good analogy. Yeah, you have the tracks, and then you have the chips that they go into, and it looks horrendous, but it's all organized, and that's sort of the way I look at the organization of all this. Right. You also have, as the cerebral hemispheres must carry out many functions, some type of intrinsic organization is necessary. Now, somatotopy is the orderly mapping of the somatic sensory system and motor system on the cerebral cortex. This organization is by function and by body area. As an example, the part of your brain, just lost the track of the mouse there. As an example, the part of your brain that controls your finger movements is right near. So, for example, here, the little fingers here is right near the place that controls your wrist movements, and that's right near the place that controls your elbow movements here. Now, the homunculus is a schematic representation of the cortical projections for motor and sensory functions. The body is drawn in proportions to the amount of cortical tissue that subsumes each part. So, for example, the sensory homunculus shown here has a prominent tongue, okay? Tongue here, and then lips kind of here. It also has and it also has a prominent hand, you can see here, but it has a small trunk, hip, and legs. Okay. Now the size of the body part is less important than the density of receptors in determining how much space is needed in the cortex in regards to this. So basically, these proportions shown, the body parts correspond to the sensory discrimination. So the density of receptors is important. The greater the density, the more cortical tissue is needed. Similar as, I won't go into the motor homunculus, not shown, and we're kind of getting a bit time-wise, but the innervation ratio is important there, where basically the number of muscle fibers innervated by one motor neuron is important, so the smaller the number, the finer control is needed, the more cortical tissue would be needed for that. Okay. Now, concerning vision, consider the above statement by the international award-winning author James Lefanu, and I'll try to speed things up by kind of reading the highlighted parts. But it now turned out that the brain created that image from the interaction of 30 or more maps within the visual cortex, each dedicated to one or the other aspect of visual image, the shapes, color, movement of the world out there. And the other thing that those take-home points is that the present one is rooted in the belief that an image of the visual world is actually constructed by the cerebral cortex. So you have 31 maps, and you have the visual world is constructed by the cerebral cortex. That's the kind of take-home message from that slide. And you have here, in this statement by Jerome Feldman, one of the most famous continuing questions in computational neuroscience is called the binding problem. In its most general form, the binding problem concerns how items that are encoded by distinct brain circuits can be combined for perception, decision, and action. For example, why don't we confuse a red circle and a blue square with a blue circle and a red square? Here you can see the parcellation or dividing up of different cortical regions involved in visual processing. So this shows a sort of, not a great picture of the brain, but this is a lateral region, the temporal lobe here, the occipital lobe, and you can see all these colored regions are involved in visual processing. This is the sort of inferior look at the brain, looking up at it, the temporal lobe and the occipital lobe here, and all these areas involved in visual processing. And so how is information from all these distinct regions, 30 or more, involved in specific aspects of visual processing, for example, shape, color, motion, depth, integrated into a unified and correct representation of the external object? Even if this could be explained, it would be like explaining how a TV converts a cable or aerial signal to an image on a screen. Amazing signal processing, to be sure, but it doesn't explain how you, the ghost in the machine or mind, is able to perceive and think about the image. There are so many unresolved questions and mysteries about the brain. Just ponder the above statement again by James Lefanu. The twin and unfathomable profundities of nature and nurture. First, how just a few thousand genes might instruct the arrangement of those billions of neurons with the hardwired faculties of language and mathematics. And second, the physical basis of that all-encompassing property of neuroplasticity, by which the brain incorporates into itself the experience of a lifetime. Let's have a brief look at language. Now, Broca's area in the left inferior frontal cortex here is usually associated with language production, that is speaking. Now, in the superior posterior temporal lobe lies Wernicke's area, shown here, which is associated with understanding language. Now, running around the lateral sulcus is in the left hemisphere of the brain. There is a sort of neural loop that is involved in both in understanding and in producing spoken language, sort of indicated here. In this loop, Broca's and Wernicke's area are connected by a large bundle of fibers called the acute fasciculus. Now, this language loop is found in the left hemisphere in about 90% of right-handed people and 70% of left-handed people, language being one of the functions that is performed asymmetrically in the brain. Surprisingly, this loop is also found at the same location in deaf persons who use sign language. Okay, now children have an innate capacity to learn language, something that adults find much more of a struggle. Electrical stimulation studies during neurosurgery have found that brain regions involved in language only approximate those indicated by older textbooks, as exact locations differ unpredictably among individuals. Many of the sites that elicit interference of language in response to stimulations or electrical stimulation are outside the classical Broca's and Wernicke's language areas. Hence, whilst Broca's area and Wernicke's area are involved in expression of speech and comprehension of language respectively, they are not the only areas involved. However, localizing aspects of language function to certain brain regions does not lessen the mystery of how it works, any more than knowing that engines power cars informs you how engines work. Now, we are told in Sapiens, A Brief History of Humankind by Yuval Noah Harari, which seems to be a very popular book read by a lot of woke people. Anyhow, I read it to review it. Anyhow, but in that book, he says that the appearance of new ways of thinking and communicating between 70,000 and 30,000 years ago constitutes the cognitive revolution. As to what caused this, the author admits to not being sure, but states the most commonly believed theory argues that accidental genetic mutations changed the inner wiring of the brains of sapiens, enabling them to think in unprecedented ways and to communicate using an altogether new type of language. We might call it the tree of knowledge mutation. Why did it occur in sapiens' DNA rather than in that of Neanderthals? It was a matter of pure chance, as far as we can tell. Now, magic bullet language genetic mutations, such as a tree of knowledge mutation being seriously espoused as responsible for our unique way of thinking and communicating, are simply not plausible to explain something so complex. To me, it's about as likely as a lightning strike hitting a car and upgrading it from a Volkswagen to a Mercedes. It's never going to happen. All right. Now, as an example of the intricate wiring of the brain circuits, consider the release of the hormone melatonin from the tiny pineal gland, the main site of its synthesis in the brain. Now, melatonin has various physiological roles, including circadian rhythm control. That is the internal body clock that tells our bodies when to sleep, rise, eat, etc. The hypothalamus of the brain, which controls both the autonomic nervous system and hormonal system, is involved in regulating the body's circadian rhythm through the circuitous connection its suprachiasmatic nucleus, abbreviated SCN, makes of the pineal gland. Okay. So the tiny SCN, or suprachiasmatic nucleus, situated near the optic chiasm, which is where sort of half the nerves of half the nerve fibers from each of the optic nerves cross over. So the SCN receives direct input from the retina's ganglionic nerve cells via the retino-hypothalamic tract, and thereby allowing eye-light exposure to synchronize the body's 24-hour internal clock. Now, the SCN controls circadian movements through connection with another hypothalamic nucleus, the paraventricular nucleus here, so PVN here. The PVN in turn signals the sympathetic, clicked something wrong there. It signals the sympathetic preganglionic nerve cells located in the thoracic spinal cord, sort of indicated here. And these sympathetic nerve cells then synapse on postganglionic nerve cells in the superior ganglion. Basically, these are part of the autonomic nervous system here. And in our autonomic nervous system, there's always two motor neurons as opposed to skeletal muscles. There's one in the autonomic nervous system. There's two, and there's a connection in the peripheral nervous system. So that happens throughout the autonomic nervous system. That there's basically a chain of two motor neurons, and a second motor neuron then projects to the gland. So from there you have the postganglionic nerve fibers, then basically innervate the pineal gland. And the pineal gland produces melatonin in darkness, but not in bright light. And so the amount of melatonin secreted is regulated by the incoming nerve stimulus, which in turn is determined by the amount of light stimulated signal received by the SCN, the suprachiasmatic nucleus. The melatonin is released into the blood, and the level of hormone in brain influences the 24-hour day-night rhythm. Now, if it all seems very complicated, then ask yourself, how could such an intricate pathway be put together except by purposeful design? The description omits all the complex interaction between neurotransmitters and receptors at the synapses, as well as the manufacture of the hormone and how the receptors of melatonin at the target nerve cells got there. Is it even feasible that natural selection sorting random mutations could put together information into the DNA to even construct one part of such a pathway, given the steps and complexity involved? All right, so not a great diagram, but anyway, I'll proceed. Getting to the end now. Neurotransmitters and receptors are themselves a world of complexity. Now, the direct effect on membrane potential at synapses by neurotransmitters is complex enough, with the opening or closing of various membrane channels, which is kind of shown in the left figure here. So usually say acetylcholine binds here, the channel opens up. Bear in mind, these channels are complicated things. There's several protein subunits, and it's all complex setups. However, there's also the even more complex indirect effects to consider, such as G protein coupled receptors, sort of shown in this illustration here. So, for example, you have a first messenger, which is the neurotransmitter, which binds to a receptor on a cell membrane. So it binds here. And this receptor is coupled to a G protein. So that's the G protein here. So here you can see here. So when the receptor binds, then the G protein activates an enzyme. So here it's the adenyl cyclase here, represented here. And this enzyme then basically converts ATP, which is the energy currency in the cell, into cyclic AMP, and it does this when after the first messenger, the neurotransmitter binds. The cyclic AMP is the second messenger, and this may open membrane channel. For example, what it may do is it may activate a protein kinase enzyme, which in turn will basically phosphorylate a substrate protein here, means it adds a phosphate group to it. And this, well, in this case, it closes a channel, okay? Now you can have various versions of these G protein coupled receptors. They may not all be as complicated, have various roles, okay? But is it feasible that such a complex system came about by chance? Okay, Now, this is my last statement by James Lafanu, which leads into a brief discussion of mind. And he says, science may have a thorough understanding of the workings of the heart and lungs, but the unpreprocessing 3 pounds of brain tissue confined within our skulls like a vast intellectual black hole absorbs the most searching forms of scientific investigation. Its most elementary currency of a single thought defies any materialist explanation, let alone how that thought might be rational rather than irrational, inspired rather than banal. Now, the brain with the mind, which I regard as distinct from the brain, allows you to reflect vividly on the past, to make informed judgments about the present, and to plan rational courses of action in the future. It endows you with a seemingly effortless ability to form pictures in your mind, to enjoy music, to dream, to dance, to fall in love, cry, and laugh. You think of your mind, but the mind is not physical. As stated by neurologist Richard Riestack, mind is not a physical structure like the brain. It is not a thing. Mind has no visible form, no aroma, no taste. It can't be held in the hand like the brain. Thoughts, the products of the mind, do not require physicality to exist. Thoughts, however, are meaningless without minds that can think and interpret them. Now, no one knows how the mind works and how it links with the physical activity of the brain. Claims that the mind and brain are products of evolution, that they are one, and that you are your brain, are simply Darwinian pledges of allegiance. The materialist or reductionist insist that everything can be reduced to matter and its movements, which in the brain means mental events such as thinking, emotions, and awareness, must break down to interactions between nerve cells and associated molecules if mind and consciousness are physically part of the brain. As the mind cannot be described in reductionist terms or reduced to matter, it ironically falsifies the very philosophy it is used to espouse. To me, it's inconceivable that the human brain, along with the non-physical mind, could have created itself by a process devoid of intelligence, plan, and purpose. It would be impossible. The human brain, including mind, does not give up its secret easily. There are so many mysteries. The thing is, if it's so horrendously functionally complex and so much is beyond their understanding, how on earth can evolution exist that there is no doubt that brain and mind are involved? In effect, they are saying that the brain is pond scum created, although they don't know how except for just so stories. In fact, one hears all sorts of just so stories about how the human brain evolved. All seem to be acceptable to some degree in academia, yet mentioned only one that makes sense, that is by the intelligent design of a creator God, and some have that as unacceptable. There's something wrong with such logic. Romans 1:22 says, professing to be wise, they became fools. Perhaps that is what is going on. Okay, that ends the talk. I'm sorry I went a bit over time of this talk.